Okay, chapter 7, all right, detection and analysis of nucleic acid. So in the previous chapter, we discussed how to purify the DNA and RNA. And now, how do we evaluate those purity and the yield, quality and quantity of the DNA and RNA? So there are different methods that we can use to determine the precise concentration of DNA and RNA. Okay, because the precise determination of their quality and quantity are important, are necessary for the downstream molecular application, like the PCR reaction and the next generation sequencing. All those molecular application require that we have a precise quality and quantity of the nucleic acid that we prepare. So I commonly use the method for the DNA concentration measurement. So basically, we can do the evaluation of the intensity of the band on an agar gel, all right? Fluorescence measurement using the various DNA binding dye, or measure the sorry, it's not shared. It's not shared. Okay, so let me see, share. So now you can see.
Yes, thank
Yes,
you.
now I can see.
Okay, so you did not miss much. This is the first slide, okay?
Yes,
This is the first slide, all right? So several methods that you can use to determine the DNA concentration: agar gel, fluorescence, UV absorbance, and the real-time PCR method. So these are 4 commonly used methods to determine the DNA concentration. And then the most convenient way to determine the concentration is the UV spectrophotometry. Okay, it's a simple DNA quantitation method, right? This is very standard technique to do the UV spectrophotometry. And in this method, basically, you evaluate. You need to have this the spectrophotometry, spectrophotometer. All right. So this is the one the picture I took from my laboratory. Right. This is a spectrophotometer, and then you need to have a cuvette, and then you put your sample in the cuvette and place into the spectrophotometer, and then through the UV light at the 260 nanometer, the absorbance at the 260 nanometer. So we call A260. And then you measure the reading, all right? Measure the reading. And so that will tell you, that can help you to determine the amount of DNA. So normally for A260, the reading of A260, every one A260 equal to 50 microgram per milliliter of the double-stranded DNA, all right? So if A260 is 1, then that'll be 50 microgram per milliliter of DNA. If it's 1 A260, that'll be 40 microgram per milliliter of RNA. All right, that's different between the DNA and RNA. All right, 50 and 40. Right, and then also from this A260 measurement, you know the quantity of DNA, and then you also need to know the quality. All right, the quality normally you need to do the ratio of 260 over the 280. Because this ratio can tell you the DNA purity in terms of the protein contamination. Because the 280, basically the absorbance at 280, that'll be the protein absorbance. That when we discuss the protein determination, concentration determination, then we will also learn the 280 that'll be used to quantitate the protein concentration. So 260 over 280 ratio that give you the DNA purity in terms of the protein contamination. And normally the pure DNA should have this ratio over 1.8 on a scale with a maximum 2.0, all right? Must be over 1.8. If the ratio is below 1.8, then you will need to do the phenylchlorophyll extraction again, right? And that's one value you need to know. Another value you need to know is A260, the A260 over A230 ratio. That's another key measure for the relative purity, because A230 will represent the presence of the chemical contaminants. Okay, so for example, the chaotropic salt, the presence of chaotropic salt, the presence of those chaotropic salt can inhibit the PCR reaction. Okay, and so 230 you used to evaluate the amount of those chemical contaminant in the sample. So the ratio, right, A260 over A230, the ratio must be greater than 1.4 on a scale of 2.0, so that we can say that DNA is pure. All right. So A260 to A280, the ratio must be over 1.8, and then A260 over A230, the ratio must be greater than 1.4, so that we will know that DNA is pure. All right. And then here, just to show the formula to convert it A260 to the DNA concentration or RNA concentration. So for double-strand DNA, so basically that'll be a 50, all right, microgram per milliliter. So the reading over 0.02, so every 1 over 0.02, that's 50. So 1 A260 with 50 microgram per milliliter. Then for the RNA, so 1 A260 over 0.025, that'll be 40 microgram per milliliter. All right, so you need to know these two numbers, 50 and 40. And this is just to show the absorbance that of the sample, different sample with different amount of the contamination. Okay, and the problem for the UV method is in the presence of the phenol contamination, you might get the false result, because you can see this the curve for the A, although it has a higher A230, it's even higher A260. All right. And then this is normal sample. Then if you have a low contamination, and then the A260 also low and much lower than this A. And then when you try to evaluate the concentration, then you mistakenly will think the C is less concentrated than A. All right. And then that's a spectrophotometer. The regular spectrophotometer is huge and big. And nowadays we have this newer development. This is called nanodrop spectrophotometer. And this is also taken, the picture taken in from my laboratory. So this is nanodrop. Each of these nanodrop is a $15,000, very expensive. And you can see there's a tip here. And that's the place where you put a one drop, like a 1 microliter or 2 microliter of sample here. Then they can determine the concentration. They can determine the A260 reading, A230 reading, and A280 reading. All right, because they use the sample surface tension. All right, and then to stay on the tip here, and so that you can evaluate the absorbance. That's nanodrop. And then the fluorometry method, basically you use the different DNA binding dye. All right, so like ethylene bromide, the standard dye, or you can use the Hosch 33258 that can bind the double-stranded DNA, and pico green. So these are different DNA dye. Then you add it, the more concentrated DNA, then you will see a higher intensity. So after, for example, here's the example, when you add a dye, and more concentrated DNA give you a more strong signal, all right? as you can see from here, the agarose gel electrophoresis. So from this very weak, and then start seeing the band 50 nanogram to 1 microgram. So the more DNA you have, more concentrated DNA you have, then more intensity you see. So that's fluorometry method. And the real-time PCR, and you can also use a real-time PCR to evaluate the concentration. But this one is just expensive and also time-consuming, but give you the absolute accurate DNA concentration. But you also need the reference curve, so that you'll be able to evaluate the amount of DNA. But the real-time PCR method we will not discuss further here, because later on we will have a chapter to discuss the PCR, and then we will discuss the PCR. And then that's the first part of the chapter is to discuss how to determine the DNA concentration. And then the second part is to discuss the separation of DNA. All right, molecular separation. Basically, the gel electrophoresis is routinely used for detection and size analysis of the nucleic acid. Right. So the separation by this method, we use the electrical current to propel the charged biomolecule, which is nucleic acid, through a porous gel matrix. Right, at the rate that is a function of the charge, size, and shape of molecule. So that means, as you can see from the panel A here, the distance migrated and the size of DNA. All right, so distance migrated will be longer when the size of DNA is smaller, right? Smaller. When the size of DNA is larger, the distance is shorter. And basically the DNA is, because it's negative charge, so you apply the DNA in the electric field, they can migrate from the negative charge to positive charge. And the separation, the distance migration, depend on the size and the conformation. So the large size of DNA will migrate slower, okay, like here 2,000 base pairs that migrate from top to the bottom. And smallest the fragment will run further, so the distance migrate is the longest, right? And then here you can see the comparison: 100 base pair marker and the 1 kb marker. So the big size DNA, they migrate shorter distance. Smaller fragment migrate longer distance. And in this curve here, to show you that the distance travel and the length of DNA, the size of DNA molecule, here give you adequate resolution, and here's a poor resolution. As you can see here, in terms of this distance migrate, here you have 500 base pair difference. But between here and here, you see that's 1 kb. If you look at here to here, the same distance, that's 2 kb to 6 kb. So that's why the resolution here is lower. Here you get a higher resolution in the bottom. So that's what this means: adequate resolution in this period, in this region, poor resolution in this region. So the agarose is a purified linear galactone hydrocolloid isolated from the agar, or the agar-bearing marine algae. It's a linear alternating copolymer of the galactose and the 3,6 anhydrous L-galactose. Okay, so that's the agarose. And agarose, and then so agarose provide this the network of a pore for the DNA fragment to go through. So that means the 2% compared to 5%. 2%, I mean less concentrated agarose. Then the pore of the network of the pore is bigger, all right? And then 5% be more concentrated. Then the pore of the network of the pore is smaller. So for 5%, the smaller fragment can penetrate the agarose easily, and then can easily separate the smaller DNA fragment. And for the lower concentration, Marina, yes.
You said for the 2% gel that it separates faster, there's more of a pull?
For 2%, because the pore is bigger, the pore size is bigger, so all the fragment, all the DNA fragment can penetrate through this faster, all right? And so it's difficult to distinguish it. The resolution is bad. They say, for example, 50 to and 200 is poor resolution. For higher concentration, then it's more restricted. Then you can see the resolution is higher for this smaller DNA fragment. Do you see the difference, right?
Yeah, but I'm
There.
just thinking that when I think of more concentrated, I would think that the reaction would like occur faster, or the
No,
DNA
no,
would.
no, because 5% is more concentrated, so the hole is smaller. The hole
Oh,
is
so there's
smaller.
basically more, there's solutes in the way, or in a way,
Yeah,
there's particles in the way.
yeah.
Okay.
And 2% the hole is bigger, but the 2% is difficult to resolve the smaller fragment. However, it's good to resolve the large fragment. Okay,
Okay.
large fragment you need a low concentrated agarose, and
Okay.
then 2%. 2% would mean a 2 gram per 100 milliliter solution, okay? And 5%,
Would be
5
5
gram per
grams.
100. Yes.
Okay.
And it's good for the smaller fragment, okay? Good.
Okay, and this is going to separate out the DNA proteins?
Protein too, but we will discuss that later, yeah.
Okay.
And the different conformation also that will be also affected because as I just mentioned, 2% you get a large hole, 5% small hole. It all depends on the size of the hole and also size of the fragment. And then if the fragment size is the same but it's different conformation, then depend on the size of the conformation, whether it's smaller or bigger. If it's smaller, then can penetrate through the hole, the pores easily. If it's a bigger conformation, it'll be difficult. So I mean, the different DNA conformation also give you different result. So for the relaxed DNA, I mean, like these, that would be on the top. They move slower because they occupy large area of large volume of the space. So that's why they're difficult to go through the pore. And the smaller one, that's a supercoiled DNA, so they can penetrate through this easily. So you can see from top to the bottom, these migrate further, and these migrate slower. And this is a standard the gel box that apparatus that people use, that we use for the electrophoresis, right? And then you apply the electric current from negative to positive. DNA migrate from negative to positive. You have agar gel in the middle so that they can push the DNA to go through the gel. And of course, you need a gel buffer, right? You need a buffer. So we have TAE buffer, TBE buffer, right? The buffer, I mean, they try to stabilize the condition during the electrophoresis so they can maintain the the pH, all right? So the pH will not fluctuate. And the TAE, that's a tree acetate EDTA. TBE is that tree sporate EDTA. TBE has a greater buffering capacity, give you a sharper resolution than the TAE. And the comp, so basically you can see here from the gel electrophoresis, you can see these are the well. This well, you need a comb and to form the well so that you can load your DNA to the well and then you perform the electrophoresis, okay? And these are typical comb that people can use. And the detection of the DNA on the agar gel. Normally, DNA is colorless. You will not be able to see the DNA on the agar gel. The reason you see this signal here, that's because you add ethidium bromide, all right? Ethidium bromide, so that means that you stain the DNA during or after the electrophoresis. But normally we add the ethidium bromide to the agar gel and also in the buffer, and so that the DNA will be labeled. And after the electrophoresis, you place the agarose gel on top of UV light, you will be able to see the DNA fragment. And another two different dye, now you can use cyber green, cyber gold, or silver stain, but the cheapest one is ethylene bromide. So this one is commonly used in the laboratory. And this is to visualize the DNA. You have ethylene bromide in the gel, and also you add to the DNA sample. After the electrophoresis, you put the gel on top of the UV light, then you'll be able to see the DNA fragment. Okay, that's the ethylene bromide. And as I mentioned, the DNA is colorless. During the electrophoresis, you will not be able to see where's your sample, where's the DNA. Okay, in order to know where is your DNA, then you need to have a tracking dye so that you can track, you can estimate where are your samples. The tracking dye, so we have two different types of tracking dye. One is called xylene cyano, and the other one is the bromophenol blue, right? Bromophenol blue normally give you like a 100 base pair, the location of the DNA at 100 base pair long. For xylene cyano will be the 700 to 800 base pair long. Okay, so that's the xylene cyano and bromophenol blue. Let me see if we can see that here. No, not in here. But if you can see here that you can see always the bromophenol blue will run in the front, and then xylene cyano later. Okay, because it will show you the large size fragment. That's a xylene cyano. Bromophenol blue will be the show fragment. Okay, so we will watch the video briefly about agarose gel electrophoresis.
Video will take you through the process.
So this is the power pack to provide the electric field, okay? And this is the gel box, all right? You're going to place the gel in the gel box.
For loading and running DNA samples on agarose gel.
See the gel box?
You will be using a mini subcell to perform agarose gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This provides for an electric current to pass, which separates the DNA fragments in the samples. First, align the gel so that the wells are closest to the negative or black electrode. DNA is negatively charged and will move from the negative electrode through the gel toward the positive or red electrode. Place the agarose gel into the gel chamber.
Agarose gel, and these are well, okay? And then you're going to pour the buffer.
Add electrophoresis running buffer to the reservoirs at each end of the gel chamber, and keep adding buffer until the wells in the gel are covered by at least 2 millimeters of buffer. Place your samples to be loaded in the correct order according to the lanes they are assigned to be run in. Check your lab protocol for this information. To obtain a DNA sample, head to the first or soft stop. Holding the plunger down, insert the tip of the pipette into the microtube with the DNA sample. Place the tip close to the bottom of the sample, then slowly release the plunger button. When loading the samples, keep the pipette tip perpendicular to the row of wells. This will reduce the risk of accidentally puncturing the wells with the tip. Lower the tip of the pipette until it breaks the surface of the buffer and is located just above or just inside the well. Slowly apply pressure to the plunger button and observe as the sample fills the well. Make certain you stop at the first stop on the pipette, pause, then slowly remove the pipette while keeping your thumb down on the plunger. Once all the samples have been loaded, avoid any bumping or movement of the gel chamber. This might result in the sample spilling into adjacent wells. Place the lid on the gel chamber with the terminals correctly positioned to the matching electrodes on the gel chamber.
Oh, by the way, we're going to run this DNA gel next week, okay?
Black to black, and red to red. Connect the electrodes to the power supply, again making certain the electrodes match the terminals on the power supply, red to red and black to black.
Black is negative, red is positive.
Switch the power supply on, then set the correct constant voltage for running the samples. See your lab protocol for this information. If a timer is available on your power supply, set the clock for the proper time for your run. Press the start button to begin the flow of current that will separate the DNA fragments. At this point, you should be able to see bubbles coming from the wires at each end of the gel box. Notice that the positive or red electrode has fewer bubbles than the negative or black electrode. In a few minutes, you should see the samples begin to migrate from the wells into the gel.
See, these
Notice
are the
as your DNA samples run,
tracking
the dye
dye.
front moves from the negative electrode towards the positive electrode.
Do you see the two band here now? Two band. That first one in the front, that's a bromophenol blue. Second one is xylene cyano. Okay. So these are the bromophenol blue tracking dye. This is xylene cyano. So that can tell you roughly the location of the DNA fragment. Okay. You don't know where are your DNA. All you have is only this tracking dye. So this tracking dye help you to determine when to stop the electrophoresis. Okay, so that's the DNA electrophoresis. And then that's for agar gel. And of course, there's another type of gel. It's called polyacromide gel electrophoresis that you use the polyacromide, right, to perform the gel electrophoresis. And for the polyacromide gel electrophoresis, it's more, you can have a higher resolution of the DNA fragment, okay? And to make this polyacromide gel is more complicated because you need to, it's a mixture of the acromide and the bisacromide. And then you need to have the polymerization catalyst, like the ammonium persulfide and the lactimate, right? So APS, ammonium persulfide, produce a free oxygen radical in the presence of TMate to drive the polymerization mechanism, okay? So that's these two are polymerization catalysts that you need to add that. And for the polyacromide gel, it's more complicated because you have two different things. You have acromide and the bisacromide. And for 6%, the 6% means 6 grams per 100 milliliter of solution. In that 6 grams of these combined acromide and bisacromide, okay, that's a combined is 6 grams. 19 to 1, that means 19 out of 20 will be acromide, and then 1 out of 20 will be bisacromide. So that would be this mean that would this what does this mean? 6%, 19 to 1. So that's 6 grams in 100 milliliter, and in that 6 grams, 19 part would be acromide, and 1 part would be bisacromide. Okay, so that's what this means. If for the polyacromide gel, you normally will see this kind of the expression. It's a high resolution. Yeah, as you can see, these are the sequencing gel. You can see these are the single nucleotide resolution. Okay. And the DGGE, that denaturing gradient geophoresis, basically, this is you use a linear increase in the gradient of denaturant. You add it to the polyacromide gel. So the DNA fragment with the same lengths, but with different DNA composition, and then they can be separated and distinguished. So the separation in the DGGE gel is based on the electrophoretic and mobility of a partially melted DNA molecule in the polyacromide gel. Okay. Even you have a DNA with the same length, but with the different DNA composition, then you can separate. So, for example, you have sample from different microbial communities, and then you isolate DNA, and then you perform the DGG gel, so that you can see, you can distinguish whether they are the same or different. And also you can identify the shared gene among different community in this case, and this unique gene within the specific community. Okay, so you have a different denaturing from 0 to 100%. Then electrophoresis this direction. Then you can see the difference: partially male to wild type, partially male to mutant. That's another way. And this one, that's a different denaturing gradient from top to the bottom, so you can separate these different fragments. That's DGG gel. And normally these are denaturing that we use, like a urea, like a 7 molar urea or 40% of formamide. Okay, that's the denaturant that can be used in a gel. And this technique we call the DGE profiling method. All right, that can be used for diagnosing the presence and the relative abundance of microorganisms, okay, in the sample obtained from the patient. All right, so that's why this is important. This is just to show the DGG gel and how they make the DGG gel. So you have the high concentration solution, low concentration, and goes through this peristaltic pump, and then the gel, and then so that give you this different increasing concentration of DGG gel. So let's watch this briefly.
Explain the role of DGG detected with this method. The reason why heteroduplex detection is interesting with DGGE is because they cannot be detected with regular electrophoresis. After this web lecture, you will understand why. DGGE is a method to observe mutations in the DNA. The principle is to denature DNA strands based on the GC and AT ratios, so basically melting temperatures. Well, you know already that a GC binding contains 3 hydrogen bonds and an AT binding contains 2 hydrogen bonds, which means that the more GC bonds in the DNA you're interested in has a higher melt temperature than a piece of DNA containing more AT bonds. Before we use DGGE, we have to perform PCR to get multiple copies of our piece of DNA you're interested in. So let's have a look. It's also important to take some controls along the process, like first of all sample at least twice, also a piece of DNA containing no mutations at all as a positive control and no DNA as a negative control. Then the samples are loaded on the gel. The cell contains the denaturing agent, which causes the DNA to melt at various stages with a melting temperature of 60 degrees. The DNA normally melts in a stepwise process, but mutated DNA strands only melts partially. This creates a dissimilarity and thus a possibility to observe DNA strands of the same length, but with various sequences. Heteroduplexes are these kind of DNA strands.
A heteroduplex is a recombination of 2 homologous DNA chromosomes, so each on both chromosomes contains
we'll skip these, right?
Which they can be commonly capital as before. These heteroduplexes are lettered. Then we have our patient sample. There are 2 blue stripes which represent our heteroduplexes when we cut at the branch point 2 and 4. As we can see, these DNA strands have a different sequence, and thus they appear differently on gel. The green stripe represents the heteroduplexes when they're being cut at branch points 1 and 3. Those sequences are equal, so 1 stripe appears. Our positive control only has 1 stripe because it does not have any mutations. Thus, it can melt completely and migrate far through the gel. And our negative control has no stripe at all because there's no DNA. Now you know that with DGGE, you can easily detect whether there is a mutation.
So, and then the community.
This is frequently used to screen complex ecosystems and analyzing various environmental samples in a short time. But also, the diagnosis of infections can be faster because of DGGE, and thus the identification of pathogens, because DGGE is not about the length, it's about what's inside that counts.
So that's a DGGE, okay. And then another one is called past field geoelectrophoresis, PFGE, all right? So basically very large piece of DNA cannot be resolved efficiently by the regular geoelectrophoresis, so we have to run this past field geoelectrophoresis, right? So basically you have a pulse of a current applied to the gel in alternating dimension to enhance the migration. And then you'll be able to resolve the larger, very large DNA fragment, like more than 20 kilobase long, okay? So for example, we have bacteria cell mixed with the mutated agarose, right? And then pour into the mound and lyse the DNA, and then you cut the DNA with restriction enzyme and then perform this PFGE electrophoresis. In the end, you will see the difference. So let's watch this video about PFGE.
Gel electrophoresis, or PFGE, is a technique used for the separation of very large DNA molecules. Under standard electrophoresis conditions, an electric field is applied in one direction on the agarose gel, and DNA molecules larger than 40 kilobases essentially move together through the gel, independent of their size. In contrast, PFGE applies an electric field that periodically changes direction. By introducing an alternating voltage gradient on a PFGE system, very large DNA fragments can be separated by their reorientation and movement at different speeds through the gel. Larger pieces of DNA are slower to align their charge to the change in direction, while smaller pieces are quicker to align, resulting in greater separation. Let's take a closer look at the equipment and conditions that will lead to successful PFGE. Here is the electrophoresis chamber, which contains the running buffer. The agarose gel should be well anchored in the middle of the chamber so that it doesn't float around. There are multiple pairs of electrodes around the gel, set in a hexagonal grid. This hexagonal shape of electrodes allows for the directional switch of voltage. The voltage is periodically switched among three directions, one that runs through the central axis of the gel, and two that run at 60-degree angles on either side. The PFGE technique takes longer than standard gel electrophoresis due to the size of the DNA fragments and because the fragments don't move in a straight line. To prevent the agarose gel from overheating due to the extended period under the electrical field, which could result in a loss of resolution, a cooling module adjusts the temperature of the buffer. A typical PFGE program is run at 14 or 15 degrees Celsius. It is important that no air is left in the tubing to and from the cooling module. On the control module, the migration protocol can be entered and monitored. This includes the initial and final switch times, the duration of the migration, the angle of electrophoresis, and the voltage. The larger the DNA fragments, the longer the pulse times should be, and the longer the total migration time. The switch time is the length of time that the electrical field is pulsed in a single direction. For example, a 10-second switch time means that the electrical field will be pulsed in one direction for 10 seconds and then switch to another directions for 10 seconds. Samples with a wide range of DNA fragment sizes, such as any of these PFG markers, can be resolved by ramping up a short switch time to a longer one over the course of the run. Most PFGE protocols are optimized for 6 volts per centimeter gel runs and optimized for a 120-degree angle. With any PFGE system, depending on the migration protocol, you can separate DNA fragments or molecules from 10 kilobases to 10 megabases. Here are separation examples of the lambda monocut mix from 1.5 to 48.5 kilobases.
So that's a PFG gel, all right? So we have different type of PFG. It depends how you arrange the gel. Okay, so for example, the one you see on the video, that's called a control clamp homogeneous electric field CHEF. So basically, you have these the from the center angle, and then 60 degree to the left, 60 degree to the right, so it's 120 degree switch, right? And then so you move the DNA to move. So basically, normally DNA molecule as large as 2 megabase pair can be separated with CHEF system, okay? And this is the field inversion FIGE. So you invert this electric field from top to the bottom and bottom to the top, okay? So it's 100 degrees separation of the electric field. So basically, FIGE can overcome the problem caused by the co-migration of nucleic acid and protein detergent complex, because this co-migration is common when both nucleic acid and protein detergent complex are larger than the threshold size. And FIGE provides a good resolution over the 800 kilobase. Another modification called asymmetry field inversion gel electrophoresis, AFIGE, is one of the modification, can be used for the detection of DNA double-stranded break. Okay, so that's FIGE. And this is the OFAGE. It's a vertical electrophoresis system. All right, so you have this the electric field that you have from A to A, and that's a 120 degree switch. Okay. Normally, it's like a 1 megabyte to 2 megabyte DNA can be separated by this method as a vertical electrophoresis method. And rotating gel electrophoresis, RGE. So basically, instead of providing the electric field different angle, they rotate the gel. So okay, between the two angles periodically. So you can separate the DNA between 50 kilobase to 6 megabase. So the application, so basically, for example, the identification of bacteria isolated from an environmental or clinical sample, and identification of the MRSA strain. So all these application for PFG. And then another topic is called capillary electrophoresis. So capillary electrophoresis is another alternative to the conventional gel electrophoresis for separation of the DNA fragment. Okay, so basically this capillary electrophoresis, they have advantage because of speed, resolution, sensitivity, and data handling. Okay, because to separate the DNA fragment only a few minutes, and also a very high resolution. Okay, and in the capillary electrophoresis CE, the DNA molecule separation usually carry out at a neutral pH. Under this system, the capillary wall are negatively charged, all right, and we can attract a positive charge counter ion from the buffer, and this attraction create an electric double layer next to the wall. So when the electric field is applied, the cation in the double layer migrate to the cathode. Therefore, the applied voltage causes the sample to migrate through the capillary and pass the detector window. Meanwhile, information is collected and stored by the appropriate data acquisition. All right, and sometimes the DNA is fluorescence label, so that when they go through this detector, they can collect the information easily. Okay. And of course, the capillary electrophoresis, not only you can use to separate DNA, you can use it to separate protein or some analytes, right? And so, for example, here we have a capillary zone electrophoresis that can separate DNA, and the capillary isoelectric focusing that separate the protein, which we will discuss in next class, all right? But here I want to show you a video, so to summarize the difference between the gel electrophoresis and the capillary electrophoresis. Okay.
So how does DNA profiling works? First, can extract DNA from the cells. Then we can use the polymerase chain reactions to produce multiple copies of specific DNA fragments using primers. The primers can be labeled with fluorescence tag, which makes it easier for us to identify specific DNA fragments afterwards. If you're interested in how PCR works, please check out the link below.
We will discuss PCR
So
later.
once
Yeah.
we have multiplied the DNA fragments with PCR, we can subject them to electrophoresis.
So let's say here they collect the DNA from the clinical sample, let's say blood, and then extract the DNA, apply the PCR, so you get different size of the fragment because the STR fragment. Remember we learned this in chapter 5, right? Short tandem repeat DNA fragment, and then they have different length, and then of course one method you can use the standard conventional gel electrophoresis to separate the DNA fragment. Okay.
The fragments get separated according to their size. By definition, electrophoresis is a separation method based on the differential rate of migration of charged species in an applied DC electric field. This simply means that the negatively charged DNA fragments will move through the medium at different speeds when we apply an electric field. This diagram here shows a typical setup of a gel electrophoresis. First, we can introduce the DNA fragments into the sample wells, and then we can apply a voltage across the gel. The negatively charged DNA fragments will be attracted towards the positively charged anode. The smaller DNA fragments can move through the gel faster, while the larger DNA fragments travel slower. The final step will be the visualization, where we can use laser to excite the fluorescence tag. The results can be visualized as a series of colors. So how does DNA?
So here, this is just this is the result of the capillary electrophoresis, because they can arrange the DNA fragment based on their size. Show the fragment go through the detector first. Okay. So all these different analytical analyte can be used in the capillary electrophoresis. Okay. So let's go through the principle of the capillary electrophoresis.
Color picks highlighting the different length.
This is the result of the capillary electrophoresis.
We can then compare the results with the DNA patterns from possible suspect. In this case, the DNA patterns from the crime scene matches quite well with the DNA patterns of suspect 2.
So this is the result from the electrophoresis and to this STR analysis, right?
Therefore, we can confirm that suspect 2 was present at the crime scene. This modern DNA profiling methods that we have shown here is a very powerful tool in forensic science, because a tiny amount of DNA sample left behind the crime scene is sufficient to identify the suspect. The DNA profiling would not be possible without the use of electrophoresis. Electrophoresis is an advanced separation method that makes many difficult analytical separation possible, from the separation of inorganic anions to the separation of DNA and RNA with unparalleled resolution. For example, to sequence DNA or RNA, we need to distinguish between long chains that may.
This we will discuss in DNA sequencing, okay?
Process, which is an even more powerful tool as compared to the conventional. We have a typical chromatogram obtained from HPLC, and on the right-hand side, we have an example of an electrophoregram obtained from CE. Now, pause this video for 1 minute. Take a pen and a piece of paper.
We'll skip
Chart
here.
places which analytical plate separation method is. Secondly, if we take a closer look at the retention time in HPLC,
So it's the better resolution. Okay, that's basically what this mean here. And now, what is this the principle of the capillary electrophoresis? Okay, here will be the principle. Okay.
The instrumentation of capillary electrophoresis is pretty simple. First, we have a few silica capillary that extend between two buffer reservoirs. Inside these buffer reservoirs, there's a platinum electrode which is connected to a high voltage power supply. Our sample can be introduced at one end of the capillary while the detector is placed near the other end. The sample valve is interchangeable with the buffer reservoir during the introduction of sample. In order to understand how capillary electrophoresis works, we need to take a look at two key mechanisms. The first mechanism is the electroosmotic flow, which governs the movement or the direction of the bulk solution in the capillary. The second mechanism is the electrophoretic mobility, which is responsible for the separations of charged species in the solution. First of all, let's take a look at the electroosmotic flow. By definition, electroosmotic flow is the movement of the separation buffer through the silica capillary as a result of the existence of a zeta potential at the solvent-silica interface. One of the keywords we see here is a solvent-silica interface. So to understand what exactly is electroosmotic flow, we need to take a look at what is going on at the solvent-silica interface. This diagram here shows the cross-section of a few silica capillary. Typically, the capillary we use in CE has an inner diameter of around 10 to 100 micrometers, with length of about 40 to 100 cm. It is usually coated with a thin layer of polyimide to protect the capillary. To give you some perspective of how small the capillary is, we can take a look at our own hair. A typical human hair has a diameter of about 100 micrometer. The inner diameter of CE capillary is even thinner than our hair. Remember the chlorophyll experiment that we have done in CM1191? We use column chromatography to separate beta-carotene and chlorophylls of the cyanonyl groups that is lower than 3. The cyanonyl groups on the silica surface are fully protonated. When the pH is greater than 9, the cyanonyl groups are almost completely deprotonated, making the silica surface negatively charged. In between pH 3 and pH 9, the cyanonyl groups are partially deprotonated. In this experiment, the buffer solution we will be using has a pH of 7.7, which is pretty close to pH 9. Therefore, it is safe to assume that the cyanonyl groups on the silica surface are dominantly deprotonated. Because of the deprotonation of the cyanonyl groups on the silica surface, the capillary surface is basically negatively charged.
All right, that's why it's negative charge.
The negatively charged surface will attract the positively charged cations from the buffer solution, forming a layer of tightly absorbed cations, which is called the fixed layer on the silica surface. However, the tightly absorbed cations can only partially neutralize the negative charge on the capillary surface. Therefore, the negatively charged surface can still attract more cations from the buffer solution. This forms a diffuse layer, where the concentration of cations is higher than the concentration of anions. Together with the fixed layer, these two layers are called the electrical double layer. At the center of the capillary tube is the bulk solution, where the concentration of the cations and anions are about the same. To complete the cross-section of a capillary, we have the diffuse layer and the fixed layer at the other end too. Let's take a closer look at what will happen when we apply a voltage across the capillary. On one end, we have the anode, which is positively charged. On the other end, we have the cathode, which is negatively charged. When the voltage is applied across the capillary, the cations in the diffuse layer will be attracted to the negatively charged cathode, dragging the bulk solutions along with them. This is what we call the electro-osmotic flow. Now, let's take a slightly more 3D perspective of an electro-osmotic flow. Imagine the capillary tube is fixed in position, and on top of the capillary wall, we have the fixed layer and the diffuse layer where the concentration of cations is higher than the concentration of anions. And then we have the bulk solutions in the middle. Imagine the diffuse layer is like a movable tube inside the capillary. When we apply a voltage across the capillary, the cations in the diffuse layer will be attracted towards the negatively charged cathode, dragging the bulk solutions along with them. That's why the electro-osmotic flow is generally towards the cathode. Electro-osmotic flow is a unique feature of capillary electrophoresis. Because the electro-osmotic flow essentially originates near the capillary walls, it has a flat flow profile, which is very different from the parabolic flow profile we usually see in case of Higgs flow.
Yeah.
Electro-osmotic flows tells us about the movement and the direction of the bulk solutions in capillary electrophoresis. And also because of the unique flat flow profile, it usually results in better separation efficiency and resolution. But it doesn't tell us about the separation of analytes. So the next question we need to ask is, how do we separate anions, cations, and so by the end of this video, we will feature species. When the voltage is applied across the capillary, the cations will be attracted to the negatively charged cathode. The anions will be attracted to the positively charged anode, while the polar neutral species remain stationary. Since neutral species are not charged, they won't be affected by the applied electric field. And there are two major factors that affect the electrophoretic mobility of a charged analyte. First of all, the electrophoretic mobility is directly proportional to the ionic charge on the analytes. So if the analytes are of the same size, the cations that are more highly charged will move faster towards the negatively charged cathode, while the anions that are more highly charged will migrate faster towards the positively charged anode. Secondly, the electrophoretic mobility is inversely proportional to the frictional retarding force of the analyte. If the analytes are of the same charge, the bulkier cation will experience a stronger frictional retarding force, therefore moving slower towards the negatively charged cathode. Similarly, the larger anions will move slower towards the positively charged anode. In addition, the frictional retarding force of an analyte not only depends on the size, but also the shapes of the analytes, as well as the viscosity of the medium. The more viscous the medium is, the slower the electrophoretic mobility. In summary, the electrophoretic mobility is directly proportional to the ionic charge on the analyte and inversely proportional to the frictional retarding factors, such as the size and shape of the analyte, as well as the viscosity of the medium. Or it simply means that the electrophoretic mobility is directly proportional to the charge to size ratio of the analytes. But there's a catch here. Since the electrophoretic mobility is related to the frictional retarding factors, the size that we are referring to here is not the ionic radius of the analyte, but the Stokes radius. So the next question is, what is Stokes radius? At the same rate as the salt packs. In this experiment, weaker hydration, the electrophoretic velocity and the electro-osmotic velocity. For the neutral species, their total velocity is equal to the electroosmotic velocity. For the cations, we can find the total velocity simply by adding the electrophoretic velocity and the electroosmotic velocity up. For the anions, take note that the electrophoretic migration is in the opposite direction of the electroosmotic flow. However, the rate of electroosmotic flow is generally greater than the rate of electrophoretic migration of individual ions. Therefore, in this case, after combining both electrophoretic mobility and electroosmotic flow, both anions, cations will move towards the cathode with different migration rate depending on their ionic charge and stalk radius. We can use the travelator as an analogy for explaining how capillary electrophoresis works. Imagine the travelator is traveling at a constant speed towards the cathode. This represents the electroosmotic flow, the flow of the bulk solution. And the cations and anions are like the little kids running on the travelator. And for some reasons, we can see this phenomenon quite often in local shopping malls. Imagine the ice cream represent the negatively charged cathode at one end of the travelator.
So, yeah, let me just
see this phenomenon quite often. Travelator run case number 5, the dianion capillary electron towards the ice cream at different.
So that's how they separate these different fragments based on the size and charge, so that you can have a better resolution. That's the the capillary electrophoresis. Okay. So that's basically the principle of the electrophoresis, capillary electrophoresis. And now